Greetings everyone. Today we're going to focus on experiments. This is a very important topic as it can be integrated with any other topic. So for you to be able to unpack those questions, you need to understand some concepts about or around the experiments. So izinto sofocasa kuzo regarding the experiments, for us to be able to unpack those questions, we have to start with the hypothesis. This will help you to understand ABCT formulate a hypothesis. Wazi into bana zintoni ekufanele uziqwalasele ukuze ube uyibhala ngohlobo lululo i-hypothesis yakho. So there must be two variables, or you must consider both variables, which is your dependent as well as independent variable. Secondly, we have to consider the relationship between those variables. Meaning, in your hypothesis, we have to talk about the relationship between the variables. Thirdly, it must be testable. It must be testable. So that is the criteria you need to consider how to formulate a hypothesis. Like an example of an hypothesis would be water has no effect on plant growth. So if you check my statement, water has no effect on plant growth, there is like water, which is an independent variable, plant growth, which is a dependent variable, meaning I've covered now variables zikhona zombini, and I'm saying water has no effect. Now I've included the relationship between the variables, that okay, amanzi akaku affecti ukukhula kwe-plants, and it must be testable, meaning I can test whether or not water has an effect on plant growth. So you should consider those three to be able to formulate your hypothesis. Then we can move to the variables when they say give them the dependent or the independent variable. Variables. Got independent, we've got your dependent. So when they talk about a variable is anything that varies or that differs. So it's actually a self-explanatory term. So xa bethetha nge-independent variable, one thing you need to know about the independent variable, it is manipulated by the experimenter. It is manipulated by the experimenter. So the experimenter will actually manipulate the independent variable, but the dependent variables depends on the independent. So your dependent variable will depend on the independent, but the independent is actually manipulated by the experimenter or the investigator. Then the aim, when they say write the aim of the experiment. The things that you need to consider, or what you need to understand about the aim. This one is easier. It is like the purpose of the experiment. You are actually telling them why this experiment was conducted. That's what they mean. So ngunobangela ba i-experiment yenziwe bebefuna ukujonga ntoni, then that's your aim. Okay, now we can move on and try to understand the experiment as well as the control. You have to know what is an experiment and what is a control. In my understanding, the experiment and the control are the same except for one factor, that the element that is being tested for, or the variable that is being tested for, is removed in the control. Umzekelo, let's say we are testing into bana amanzi anayo na effect on the growth of the pine trees. Then the experiment would be pine trees ezizo nkcenkceshelwa ngamanzi, for example. Then the control will be ezi ezingazunkcenkceshelwa ngamanzi, so that they will be able to compare umohluko between eziwafumene amanzi nezingawafumananga. That's our experiment as well as our control. So the experiment is the same as a control except for one factor, which is like the variable that is being tested for is removed in the control group. That is the only difference between our experiment as well as our control. Otherwise, everything remains the same, because the type of the pine tree, the environment, and everything will be the same. The only thing that will be different is that in the control, we will remove the variable that we are testing for. Then in the experiment, we won't remove it. So in summary, the experiment is the same as the control, but the difference is that in control, we remove the independent variable. Actually, yes, we remove the independent variable, because water in that case is the independent variable. So we would actually deprive the control group of water, so that we can see the difference between the pine trees that were watered as well as the pine trees that were not. Those that were watered, experiment. Those that were not, control. Why control? Because they are deprived of the independent variable, which is water. Hopefully that is making sense. So the last part of this lesson would be the importance of the control in the investigation or in the experiment. Like, why is the control important? So, control is important because we want to make sure into bana results zethu are not random events. That means whatever esi-observileyo ibingazukwenzeka if bese remove i-factor ethile. To be clear enough, sizama ujonga into bana ukukhula kwe-pine trees kuxhomekekile emanzini na. So we need to have a control so that kwi-control sizowa-remove amanzi so that sizobona uba okay le nto yenzekileyo kwii-pine trees eziwafumene amanzi izokwenzeka nyhani na nakwii-pine trees ezingenawo amanzi. Because if ibingekho i-control, that means ezi ezingenawo amanzi, we will never be sure into yobana i-effect esiyibona kwii-pine trees yenziwe ngamanzi na or ibizokwenzeka regardless of bana amanzi ebekhona okanye bengekho. So that's the main reason why we have a control, so that we make sure that our results are not just random events but are actually the result of whatever we were testing for. I hope that that is clear, but if it's not clear enough, we will chat about it and we will continue in the second part of the same topic. Thank you.